Hi there, this is 7-5 Codominance. Codominant is just another pattern of inheritance in which you are not completely dominant, you're not dominant or recessive, you're not incompletely dominant, you're not an in-between of the two, but you're codominant, which means I get to see both the dominant and the recessive at the same time. So I've got these two cats right here. This one showing a very good example of codominance, where I get to see one half of the cat seems to be a black cat with green eyes, and over here we have another cat, which is an orange cat with blue eyes. So what's happening in this particular case is that the black isn't dominant over orange for codominance. For complete dominance. The black is not mixed with the orange to be incomplete dominance, but now we have black is showing next to orange, which is codominance. So codominance is just when two alleles like to work together to produce a phenotype that shows both traits equally. And so here is a rather silly example, but kind of shows the idea that we have a duck with a duckbill and a beaver with a beaver tail. And when you mix them together, you still see both traits equally, and you get a new animal, a platypus. Now, that's not really the case. Platypus is its own individual animal, separate from all these guys, and but the idea is kind of the same. And this picture, I like this one because it shows it very well. If these goats had a big B, which represented the black, and yes, once again, my pen decides to do its own thing. And I have a little b that represents, come on now, that represents white. That's, I need a new one. Anybody want to buy me a new one? Come on now, come on, make a tea. Yeah, whatever. So if I have a big B which represents black and a little b which represents white, if these goats were showing complete dominance, big B would be black. Big B, little b would be black, and two little b's would be white. So there's only two choices, a black and a white goat. If this were showing incomplete dominance, big B would be a black goat, big B, little b would be a gray goat, and little b, don't even think about a cat, would be a white goat. But in codominance, what happens is the heterozygote, get it? Heterozygote. What happens there is that big B is black, big B is black and white, and little b is white. So the heterozygote happens to be a little bit of both. Now it could be where the heterozygote shows the little bit of each, a little bit of big B, little b. But it could also be like on our flowers too, where it could be two different dominant alleles, and so the big R, which was red, and the big W, which was white, instead of being a pink flower, would just be a red and white flower. So here is an example of that exact case. This is called a Camellia Flower, and what happens is we have big R's which represent red, big W's which represent white. Now if you have two big R's together, you get a red flower. If you have two big W's together, you have a white flower. But if you have a big R and a big W, well, you don't get a pink flower, but you end up with a red and white flower, which we call codominance because the red and the white are working together like co-workers. They're each individually being seen. Now, this doesn't very much happen in humans. What typically happens, or the one main example that we have in humans, is blood typing. You have 4 different blood types, A, B, AB, and O. And the A type blood has an A protein represented by a triangle here on the surface of their blood. The B blood type has a B protein on the surface of their blood. The AB blood type has an A and a B protein on their blood, and O's have no proteins in their blood whatsoever. Now, whatever blood type you have, you also have these things called antibodies, which I kind of think of them as little Pac-Mans because they go around seek and destroy things. And they look for things that don't quite fit to your blood cells. So if you have a type A blood, you should not have any of these B proteins around. So this guy will go around and search for those B proteins. And if he finds one, he'll kill them, and that causes a blood clot, which could eventually kill you. So if you have type A blood, you can't get B or AB because it'll kill you. If you have type B blood, you have anti-A's, so I can't get A or AB. If I have type AB blood, well then it doesn't matter because I don't have any antibodies for A or B, or else I'd kill myself with the A and the B, so there's no antibodies there. And if you have type O blood, well, you have anti-A's and anti-B's, So you can't get anything but type O blood. So to figure out your genotype and phenotype of your blood type, it all depends on this thing called codominance. Now there are two different types of alleles. This is a capital I. Right there, it looks like a big L. Capital I is the dominant form of the gene. Little I is the recessive form of the gene. So just like before. The only difference now is that the capital I, the dominant, comes in two varieties. We have an A variety and a B variety. So we represent that by just tagging along the letter in the upper right hand corner of the big I. So if I have a dominant A, I represent it with a big I and an A. If I have a dominant B, I represent it with a big I and a B. Now the little I doesn't have an A or a B, so he just goes everywhere by himself. Now if I have type A blood, I can have two different genotypes for type A. I can be homozygous, which means I have two dominant alleles, each carrying with them the A protein. Or I can be heterozygous. I can have the dominant A and a little recessive. And so the A still is dominant to the I, the little I, which means I still have type A blood. Same thing for type B. I could be homozygous B, which is two big I's and two big B's. Or I could be heterozygous B, which is a dominant I with a B and then a little I. Now the only ones that only have one variety, or one genotype, would be AB and O. Now AB, there's two dominant alleles. So it's homozygous dominant, but I have a dominant A and a dominant B. Since there is no in-between of A and B, I can't be incomplete because there's no blending between the two. But what happens is in my blood type, I show both A and B equally. So since they're working together like co-workers, I would say that AB blood is codominant, that the A and the B are codominant to each other. One doesn't win out over the other. When I have an A and a B, I'm not type A, I'm not type B, I'm AB. So they work together. Now, if I have nothing but the two little recessive guys here, there's no A or B to dominate over them, so I get to see the recessive. Well, the recessive is O. So if you have type O blood, that means you have no dominant A's or B's, and all you have is little bitty I's. Okay? So, let's see, I didn't have another page. So you can do Punnett squares just like you normally would with any of the genotypes. You just make sure that there has to be two I's, either two big I's, a big I and a little I, or two little I's. So just like before, you need a big letter and a small letter. And then the only thing that's different is that attached to the I's will be either a big A or a big B hanging along with the capital I. But remember, no A's or B's tag along to the little I's. Okay? So in humans, this is one of the main traits that we have that shows codominance. Alright, next thing we're going to take a look at is polygenic traits and multiple alleles. Alright, see you later. Bye-bye.